Uh, Hello, everyone. Today we're going to give a short lecture on the bioproduction of terpenoids. The content of this lecture includes 4 parts, which are introduction, precursor pathways, representatives, and future perspectives. First, let's have a look on what terpenoids are. Terpenoids are a large and diverse class of naturally occurring organic chemicals derived from the 5-carbon compound isoprene. The terpenoids contain the isoprene polymers and their oxygenated derivatives. Terpenoids are the largest class of plant secondary metabolites, representing about 60% of known natural products. They are widely distributed in all kinds of fruits and vegetables that are pervasive in our daily life. Based on the number of the isoprene units, terpenoids can be classified into several groups. Here is a table showing the name and the molecule structure of typical examples of terpenoid species that have 1 to 8 and over 8 units. Terpenoids not only have a great diversity, but they can also provide great values for humans. Most terpenoids have strong aroma and volatility, so they are often used in the manufacture of essence and edible flavors. Hence, they are indispensable raw materials for the cosmetics and food industries. Some terpenoids are the main active ingredients of medicinal plants and have extremely high medical value. For example, artemisinic acid is well known for its antimalarial activity. Limonene can be used to treat gallstones. On the other hand, because terpenoids generally have toxicity, they are also widely used in the manufacture of insecticides and antibacterial products. In addition, due to their high combustion heating value, low freezing point, and a good fluidity at low temperatures, some terpenes, such as alpha-pinene, have become potential fuel additives that provides broad prospects for the development of biofuels. Next, we're going to explain why we focus on using microorganisms to produce terpenoids. The traditional methods of acquisition mainly include direct extraction and purification from plants and cell tissue culture. The direct extraction method is currently a common production method. For example, extracting essential oils from the peels of citrus, lemon, and other plants. The plant cell tissue culture have developed rapidly in recent years. However, due to the long growth cycle of plants, the limited content of terpenoids synthesized by the tissue cells, and the large loss in the process of extraction and purification, terpenoids production by the above method is poor in purity and yield. In addition to these 2 methods, there are also studies on chemical synthesis methods. However, due to the complexity of the terpenoid synthesis pathways and unfavorable factors such as optical isomers of the products, this method is not feasible in industrial production. Fortunately, in recent years, rapid development of modern biotechnology have provided new ideas for the synthesis of natural products. Synthetic biology and metabolic engineering make it possible for people to construct heterologous synthetic pathways of terpenoid compounds in microorganisms and use the metabolism of microorganisms to synthesize terpenoids. The basic idea of this method is to collect genes from enzymes in different terpenoid synthesis pathways, make a series of modifications and optimizations, and then introduce them into a suitable host to use their metabolism for production. There are 2 known biosynthetic pathways of the precursor IPP and DMAPP, namely the MVA pathway and the MEP pathway. The MVA pathway mainly exists in the cytoplasm of higher plants, and they also find in some archaea, fungi, and other eukaryotes, while the MEP pathway mainly exists in the protoplast of bacteria, algae. In the MVE pathways, 3 molecules of acyl-CoA are condensed to form HMG-CoA. HMG-CoA is further reduced to produce mevalonate. Then mevalonate undergoes phosphorylation and decarboxylation to become IPP and high. In the MEP pathway, G3P is condensed with pyruvate to form DXP, and DXP is catalyzed by reductive isomerase from MEP, and then undergoes a series of phosphorylation, cyclization, and dehydration steps to generate IPP. IPP isomerase catalyzes the conversion between IPP and DMAPP, which are, as general precursor, play a very important role in the synthesis of all kinds of terpenoids. After the precursors are generated, they can go through various downstream pathways to become distinct terpenoids products. Next, we're going to introduce synthetic pathways and some advanced researches of 3 representatives, which are atominic acid, lycopene, and retinol.
Okay, now I'm going to introduce the retinoids. Retinoids are required for survival of mammals and are needed to support vision, normal cell differentiation, and proliferation, embryonic development, normal immune function, and reproduction. And the most famous retinoids, vitamin A, also called all-trans retinol, is considered the most multifunctional vitamin in human body. And there are 4 generations of retinoids. Here is a picture of chemical structures of several retinoids. And next, I'm going to introduce the development of retinoids. Here is a picture of retinoids, beta carotene content in chosen fruits and vegetables. So it is easy to say that the oldest way to obtain carotenoids is extraction from plant material based on physical-chemical processes. They are extracted from green parts of plants, flowers, fruits, seeds, roots, and tubers. The main disadvantage of the production of carotenoids from plant material is the high cost, geographical determinants, and the seasonality of the raw material. And chemical synthesis have been used in carotenoids production since it was developed in 1950. Among all known methods of obtaining this production, the microbial biosynthesis is of particular interest. The need for commercial production of nitro pigment, including beta-carotene, has led to intensive research in this area. On the beta-carotene production industry scale, cultures are conducted in 3 main stages: inocular multiplication, biomass biosynthesis, and the carotenoids extraction. Depending on the final form of beta-carotene, biomass is subjected to appropriate technological operations. The beta-carotene biosynthesis rate depends on the size of the tank and decreases with capacity extension. The beta-carotene acylation process is determined by product's commercial form. To date, there is no commercially viable yeast-based beta-carotene production method. The necessary condition for the development of such technology is to know the mechanisms of its biosynthesis by yeast characterized by high rate of beta-carotene biosynthesis, biomass growth, and presence of other valuable compounds, increasing the quality of yeast biomass and creating opportunities for use in various branches of industry. And next, I'm going to talk about the pathway to create beta-carotene. And as my group members mentioned, there are 2 ways to synthesis beta carotene, the MEP pathway and the MVA pathway. And although much progress in beta carotene production using the MEP pathway has been made, this approach is still suffering from limitations due to the regulatory mechanisms present in the native host. To improve beta carotene production, the optimized MEP and MVA pathways were co-expressed in E. coli. And from this table, we can see it has 113-fold higher production than the native MEP pathway. So the result shows that co-expression of the optimized MEP and the MVA pathways in engineered E. coli could result in a considerable increase in beta carotene production. And next, I'm going to introduce the conversion of beta carotene to retinoids. Retinol is converted to retinol and the retinoic acid by retinol dehydrogenase and retinol dehydrogenase oxidase, respectively. And the retinol will is esterified to retinol esters by retinol alkyl transferase. And then my group member will introduce another representative.
Here I will introduce the artemisinic acid. Artemisinic acid is the precursor for artemisinin. It is the semi-synthesis of artemisinin from microbes, sourced precursor artemisinic acid. It could be a cost-effective method, environmentally friendly, and it have high quality and reliable source. Besides, according to some research, artemisinic acid also have those activities of anti-malarial activity, anti-tumor activity, and antibacterial activity. This can make them have many medical applications. In synthetic biology, the reconstruction of a complete plant terpenoid pathway in cells such as E. coli and some yeasts, there are novel avenues for the production of artemisinic acid, especially our yeast. Here is recognized as a better platform to synthesize artemisinic acid, and it has the yield of artemisinic acid and has been achieved up to 12.5 grams per liter. And then the conversion of the acid to artemisinin has to be processed with the aid of chemical methods. Here, yeast uses MVA pathway as the substrate to produce terpenoids. In the process of the heterogeneous biosynthesis routes of artemisinic acid connecting with FPP, the regulation targets include homofentanil synthesis, cytochrome monooxygenase, artemisinic alcohol dehydrogenase, and artemisinic aldehyde dehydrogenase. That's the whole MVA pathway in for the artemisinic acid production. And next, I will summarize some. Some ways to improve the yield of the acid. The first one is to discover and engineer some enzyme. For example, increasing the content of enzyme while the expression of fusion protein is an effective way. Besides, metallic metabolic flux and metabolic pathway analysis can also be applied to evaluate the cell condition and reveal the key factors affecting the end product production. Finally, feedback regulation and contrast new platform based on inverse metabolic engineering are also promising approaches to improve the yield of the acid. That's a brief introduction about artemisinic acid. Let's invite another group to introduce.
I will have a brief introduction about the lycopene. At first, the lycopene has many applications. For example, it is widely used in food additives and drugs. Also, it has some health preventive effects. For example, it can be used to heal some lung cancer, and also it's a good pigment in red because it's very natural. And because of its wide application, so it has a huge market. According to some data, the global market for the lycopene was about 84 million by 2018. And also, its ingredient that is tomato, it has about 160 million tons per year. So we have a great requirement for the lycopene. So there are 3 different ways to product it. The first one is about the extraction from some plants, mainly from the tomato, but it has the disadvantages of it requires huge amount of raw materials with a low yield, and it also requires complex process and require rigid condition. The second one is chemical synthesis, but it will faces the problems of producing some chemical residue. Also may be very sensitive to heat and oxygen. Because of the disadvantages of the 2 methods, microbial fermentation comes into and it's good in it is a cost effective, environmental friendly, and also very sustainable. And next, I will have a brief introduction about the bioproduction, about how to produce lycopene. I'm showing the figure on the left-hand side after the MEP pathway and MV pathway, and the precursor will go into the lycopene pathway, and with the CRT enzymes, finally it will produce the lycopene. And in order to, there are some general approaches to improve production. The first one is the optimization of the MEP pathway. In this method, it may be mainly focused on releasing some active burden. And the second method is about CRT genes overexpression, because there are many CRT enzymes used in the process, and by this way we can increase the catalytic activity and host capability of enzymes. Also can relieve the rate limiting step. The last one is the cofactor supporting. For example, it can be used. It can increase the precursor G3P and improve pyruvate for MEP process. Also can improve the acetyl A supply for MV pathway. And next will be the perspective of our topic.
So through the literature review, we concluded that the main bottlenecks in the current research are following points: the cytotoxic mechanism and the effects of terpenoids on cell growth are still elusive. Determination of the tolerance genes and regulation of their expression together with optimization of key enzymes and improvement of their poor function are challenging due to the introduction of exogenous pathways. Insufficient flux of precursors such as IPP, DMAPP, and GPP cause difficulties to achieve industrial level output. It is also necessary to consider the adaptation relationship between the metabolic pathways and the host, and systematically optimization from a global perspective can be difficult. Next, in response to the above bottlenecks, many scholars have been actively studying in recent years, and with their efforts in the near future, we expect to see the combination of chassis modification and pathway modification. Advances in metabolomics and proteomics, such as directed evolution of enzymes and rational design of proteins, can help to identify bottlenecks in the synthesis pathway more quickly. New computational tools and increasingly powerful gene editing technologies can help us to quickly introduce and assemble mutants into high-performance chassis strains tailored for the industrial production. The application of new methods in the construction and optimization of microbial factories will greatly shorten the distance from the laboratory to industrial production. With the continuous breakthrough of bottlenecks and limiting factors, we look forward to realizing of industrialized production of terpenoids by microbial synthesis and make our life better. That's all of our lecture. Thanks for your attention.